So good morning. We're going to start this second day. Co-chair with me is Luisa Bracci.
Good morning.
Luisa Bracci. Let's start, as usual, with this session related to innovative pharmacological approaches with market and trends brief insight from Andrea Fraticelli Malizia from UNIP. Please, Andrea, thank you.
Good morning everybody, and thank you very much, Andrea, for the nice introduction, and for Francesco for inviting me today to open the second day of session and works at these event acts. So today, Today we are going to just give a very preliminary introduction about the pharmaceutical therapies. As you can see here on the right part of the slide, you can see that mainly the different kinds of therapeutics can be divided into two different major groups: large molecules and small molecules. As you can see, there are almost 50% for each one. But you can see the small molecules are called more conventional drugs, meaning actually there's not really much to be done in a matter of innovation, except for a few stuff about computational chemistry and manufacturing. But instead, I like to focus on the 44% for the large molecules. These include several different kinds of therapies, completely new therapies, like cell therapy, gene therapy, monoclonal antibodies, mRNA therapeutics, and antibiotic resistance. So we are going to dig deeper in each single one in the next few slides. So I want you to focus more on the market value and the center of the slide than all the other information you can see on the side. That's because cell therapy has been for so long in this market, in the R&D, that recently they switched completely strategy. Instead of using cell therapy as a potential replacement therapy, they started using cell therapy more from an inflammatory point of view, and especially you might have heard recently about the CAR T cells. Now that's why actually we have a double-digit CAGR, composite annual growth rate, of 22.67%. That's because we're focusing in the next 10 years on big advancement in this field, and Novartis is definitely one of the top and the lead in this market. Gene therapy, you might have heard about the gene editing therapies, but that's not only. We're speaking about CRISPR, Cas9 therapies, and so on, so on. So even in this case, it's almost double digit, almost 20%, but here you can see it's almost 19%. The main one is Roche, and the second one emerging is CRISPR, but you can name it several different kind of competitor CRISPRs. In this case, we might have Intellia, Editas, in the CRISPR-Cas9. So, but unfortunately for these such innovative therapies, you see that there are some kind of pitfalls or drawbacks that the people are facing the R&D, and especially right now, likely in the clinical studies, including a potential oncogenic activity by these new technologies. But luckily there's a lot of hope for the people who still suffer of genetic mutations, as you can see an example in cancer or even the muscular disease area. So that's why we're switching to the next topic, which is monoclonal antibodies. Monoclonal antibodies has been for so long in the therapeutic market, but still right now in 2024 we are facing a quite challenge to diversify this kind of therapeutics to make more precise, less immunoreactive. So that's why actually people are focusing on CAGR 12.4 for monoclonal antibodies. That's the old school fashion style of developing the antibodies. But you can see on the right side instead for the ADC, the antibody drug conjugate, that's something that people are developing right now in the R&D, so they are right now entering in the clinical studies, and we hopefully to see a lot of results coming, especially from the main sector, from the main leader Pfizer, and coming after immediately Immunogen here in Europe with an amazing platform therapy. For The next one, mRNA therapeutics. Very likely all the people attending today have heard about the Moderna, BioNTech, all the different vaccines during the COVID time, the COVID-19. So right now, these companies are facing a huge problem moving forward and developing something more than just vaccine. So just recently, it's the news of a few days ago, Moderna reshaped completely its pipeline, moving out from COVID, a different kind of virus infection diseases, but unfortunately they cut down the different kind of therapies as a strategy point of view for the pipeline, cutting down all the oncology. That's because mRNA techniques is absolutely great, amazing new technique. We actually have shown very amazing results during the COVID time, but unfortunately it's facing several different kind of problems. An example for the cancer, moving through the microenvironment, which actually is unfortunately very challenging for these molecules. So, and for the antibiotic resistance, that's something unfortunate that we start facing the last 50, 70 years. The bacteria start creating a new resistance to normal antibiotics, so we need to create new antibiotics, and so far actually been pretty challenging, but unfortunately new bacteria, new antibiotic resistance came out, so we need to keep track of these infections. So that's actually specifically in this field, Merck is receiving a lot, and I say a lot of grants from government as well, because it's the social burden as well, having a kind of infection from antibiotic-resistant bacteria. So I want you to focus more on the top part of the slide and the bottom. I mean, actually, these are the tech trends and socioeconomic impact, specifically speaking for the pharmaceutical therapies. So you can see on the left, the antibody and oligonucleotide conjugate. So we're not speaking anymore about just antibody and oligos. We are combining these two together. That's actually where the R&D is moving forward and trying to advance as much as possible. We're speaking about oncolytic virus therapies as well as being used for gene editing technology as well. The dual targeted ADC, as I showed before in the previous slide, the gene therapies using lentiviral vectors, gene therapy is absolutely great, amazing, has shown pretty amazing results in preclinical and in vivo animal models, but unfortunately combined with the lentiviral vectors, that's because we're facing a major problem about the delivery itself. And therapeutics exosome, with several pros and cons, because for the exosome has been shown to be amazing particles containing biomarkers, and so that's absolutely used for potential strategy. People are still involved on creating new exosome as a potential therapeutics. So this is the last slide. So just focusing what actually as an innovation process, the therapeutics and the pharmaceutical market is facing from now on, for at least for the next 5-10 years. First of all, it's about the off-target effects in CRISPR and base editing pose safety concerns, including cancer risk. That's why CRISPR is the leading company right now in the CRISPR-Cas9 technology. And they are start re-challenging themselves in the phase 2 for their product, because the FDA and the EMA are looking very carefully and deeply on the potential oncogenic effect. The second point is about delivery challenging, especially in solid tumor, limited mRNA therapies beyond vaccine. That's why actually I told you about the Moderna. So they are shipping completely their pipeline. Their stock price are completely down almost 200% from the last few weeks. That's because the cancer is still quite untreatable, at least for these kind of technologies, for the mRNA technology, due to the different microenvironment effect. The third one is complex manufacturing process hinders scaling of ADC, exosome, and viral therapies. That's because we are speaking about large molecules. We're not speaking about small molecules. Small molecules are pretty easy and straightforward, let's call it this way, for the CMC, for manufacturing. Here in this case, instead, we're speaking for something very challenging, even for the manufacturing facilities. So it's not anymore that you knock at the door of the CDMO asking for a potential service. Even the CDMO itself, and even in some cases, even the regulatory agencies like FDA and the EMA, they face challenges on acquiring new information about new technologies. And definitely AI. It's kind of buzzword at this meeting, but the lack of digital infrastructure delays AI adoption in pharmaceutical development. We have seen that in the R&D in the pharmaceutical market, we are collecting a lot of information, but we don't know very much to do what to do with this kind of information at the end. So that's why combining artificial intelligence with such enormous data source would be very helpful for not the company itself, but for all the market and all the pharmaceutical companies as well, with immediately results in the society as well. Thank you very much.
Oh, thank you very much for this extremely interesting presentation. We have time for one question. Do we have? Okay, may I ask one? No, just one. I was waiting for. The question is if you think of all these hindrances and all these difficulties and many possible barriers that you illustrated, how do you see the possible development for rare diseases? Because in those cases, the economical value is something that may somehow impede the development.
So based on my experience, because actually I was working in the neurodevelopmental area, including some rare disorders like Rett syndrome and Fragile X syndrome, I can say from the regulatory standpoint of view, the FDA and the EMA, including, they try to facilitate as much as possible, but anyway, without creating any kind of burden for the society and especially for the patients. They try to facilitate the advancement of the new technologies toward the preclinical and clinical IND-enabling studies and the clinical studies as well, providing the fast-track designation, fast track designation, and so on, trying to, in some cases, even provide grants to support the research. And that's more for the regulatory standpoint view. From the R&D, unfortunately, I can say, in working in academia right now, that thanks to institutions like Teleton or even the European and the worldwide organization, including sometimes organization of patients, it's a way to communicate and share data and scientific knowledge with different research groups around the world, because unfortunately there's not many academic research groups within the same university and sometimes even in the same country. So that's better to share the common knowledge and scientific knowledge with all around the people worldwide.
Thank you very much. So if we don't have other question, we can proceed to the next speaker, who will be Claudio Suporan from the University of Florence, and he will speak about drug design of potential enzyme inhibitor-based drugs for neurodegenerative diseases. Thank you.
Thank you very much for inviting me in this exciting meeting. I am part of Spoke 8 of Tuscany Health Ecosystem, and I will try to show you the results that we obtained up until now. After the very nice introduction from my colleague, I would say that in the human genome we have around 2,000 genes, which means we can have 2,000 or more proteins, considering also the alternative splicing. And we expect quite a large number of potential drug targets, considering both proteins, nucleic acids, and so on and so forth. However, unexpectedly, the number of drug targets of all approved drugs is quite low. In this analysis of 2006, you may see that 324 drug targets were known, which account both for the human genes and the genes of our pathogens. In the 18 years that passed, the situation is not dramatically changed because we have around 400 drug targets. This means that we may have quite a lot of space for innovative drugs. We should also consider that most of the drug targets that are used nowadays are enzymes. Around 50% of them are enzymes, around 30% are G protein-coupled receptors, 7% ion channels, and so on and so forth. Enzymes, well, I make this very small introduction for people who eventually are not active in drug design. Enzymes, we know they are biological catalysts. They are involved in most metabolic and physiological processes. Imbalances in their activities, either up or down regulation, cause diseases. And of course, we can act with inhibitors or activators of enzymes to correct such defects. And there are many examples of widely used drugs which are, in fact, enzyme inhibitors, like the painkillers like aspirin, ibuprofen, indomethacin, and so on and so forth. Most anti-infectives, antibiotics, antivirals, antifungals also belong to the enzyme inhibitors. Agents for the treatment of high blood pressure, phosphodiesterase 5, and I could continue with very many other examples, but also many anti-cancer drugs belong to this. So we are involved in the neurodegenerative diseases only in the last several years. We know that there are different diseases which are characterized by protein misfolding. The most relevant one is Alzheimer, on which I will speak, but it's not the only one. There are also diseases like Huntington's, Parkinson's, the prion disease, and so on and so forth. Usually there is an issue with the aggregation of proteins. Normally the misfolded or badly folded, unfolded proteins are recognized and they are degraded at the proteasome level, but there are situations in which they accumulate, and this leads to problems, to health problems. And the most investigated one is, of course, Alzheimer's disease. It's a disease with few therapeutic options nowadays. Even if some monoclonal antibodies have been approved recently, their efficacy is rather limited. We know that it's an irreversible progressive brain disease which destroys memory and thinking skills. And what it's typical for this disease is these structures. These two structures that I show here, one it's called beta amyloid plaques. They are formed outside the cell and they contain a protein called amyloid beta with other materials, cell material debris and similar things which accumulate around the cells. Whereas the second structure, it's called neurofibrillary tangles. They are formed inside the neurons. They are twisted fibers that build up inside these nerve cells, and both of them interfere with the normal function of the brain. Alzheimer's disease is characterized also by other abnormalities, for example, in the neurotransmitter system. The most affected one is the cholinergic and glutamatergic transmission, but also there are some reduction in the monoamine neurotransmitters, that of some peptides, whereas the inhibitory, the GABA neurotransmission is not much affected in these conditions. Excuse me. And obviously, being such a terrible disease with few therapeutic options, there are many drug targets which have been proposed over the years. They include some enzymes like the cholinesterases, the monoamine oxidases, some neurotransmitter receptors, and the mitochondrial damage, and so on and so forth. You see quite a lot of them, but none of them, unfortunately, works well for the moment. A few years ago, in collaboration with colleagues from my department and an Italian-American scientist, she's originally from Florence, but she's active at Temple University in USA, we proposed carbonic anhydrases as drug targets for Alzheimer's, and I will show you why we have made this proposal. So there are clinical data which show that carbonic anhydrase is downregulated in the brain of many patients. So there are data in post-mortem patients for which the levels were measured. The enzyme which is present was also shown to be nitrated or oxidized. This leads to impairment of its functioning. And also the plasma levels in some patients still living with this disease were showing that there is a decreased, I think there is some noise probably from outside. Anyhow. So this is one of the reasons why we investigated carbonic anhydrases in Alzheimer's disease. The second issue is that in the last 10 years, we show that carbonic anhydrase is involved in modulation of memory. There are implications for the treatment of many cognitive disorders. So there is work which we have done with the group of Patrizio Blandina, Gustavo Provensi, in which we show that the carbonic anhydrase activation is important for the recognition memory, of course in animal models, for the extinction of fear memory, and so on and so forth. So there are strong connections. But what is carbonic anhydrase? Well, these are enzymes on which we worked for quite some years. They catalyzed a very simple reaction, the hydration of carbon dioxide with formation of bicarbonate and protons. It's a real simple reaction. You may ask, why do we need that? Because it's very slow at physiological pH. The enzyme, it's catalyzing efficiently. We have 7 orders of magnitude an increase in the formation of bicarbonate and protons in the presence of the enzyme, which explains why there are 8 genetically distinct families encoding for them in organisms all over the phylogenetic tree. Clearly, from 2 neutral molecules, you obtain a weak base, bicarbonate, and a strong acid, the protons, and this means that the enzyme is mainly involved in pH regulation, but not only, it's also involved in metabolism. We know in detail the catalytic mechanism of all these enzymes, so we work with representative of all 8 genetic families. We know the active site architectures, but I will not enter into too many details. In humans, there are only alpha carbonic anhydrases of the 8 genetic families I mentioned, but there are actually 15 different isoforms, which is nice from the point of view of investigating them, but it's also complicated for medicinal chemists because we have to design inhibitors and activators which are selective for them. You may see that they have different localization within the cell with cytosolic, mitochondrial, membrane-bound, and secreted enzymes. They have different activities, and they are involved in different diseases, but I will not enter into too many details here. So in the brain, we have 11 of the 14 isoforms, and we don't know why we have so many isoforms. We hope to determine this with the help of this project in which we are involved. And clearly in the central nervous system, they are involved in pH control of the brain, which is a tightly controlled process as in all organs, in excitability of the neurons, in the production of cerebrospinal fluid, and also in the regulation of cerebral blood flow. In fact, inhibitors of these enzymes are in clinical use for decades as antiepileptics for the management of idiopathic intracranial hypertension and for other neurological conditions. But what we are interested in are the activators, because as I mentioned, they are involved in cognition. And in fact, in the late 90s, it was shown by a colleague from Finland that in the presence of the activation, the GABAergic transmission, the GABA, we know very well, is the inhibitory neurotransmitter, becomes excitatory, which may explain why we have an enhancement of the cognition in the presence of the activation of carbonic anhydrase. Of course, the mechanisms are a bit more complicated, but just to simplify them, I show that. And the pharmacology of the activators is also rather complicated. I will not mention everything, but in 2017, with the group of Patrizio Blandina, we showed that the activators which were administered to an animal model helped an enhancement of the object recognition memory. And we also found how this occurs through the activation of a kinase, more precisely the ERK kinase, in the cortex and hippocampus of these animals. Later, we also showed with the same group and the group from Brazil that the activators are enhancing the consolidation of the fear extinction memory, which may lead to interesting opportunities for the pharmacological treatment of diseases for which there is not an appropriate treatment. In the project in which we are involved in Spoke 8 of THE, we proposed a design strategy of modulators of the enzyme activity within the brain, using both activators and inhibitors of the carbonic anhydrases, which were hybridized with different other chemotypes, for example, cholinesterase inhibitors, monoamine oxidase inhibitors, and so on and so forth. So of course, the chemistry is nice, but I will not enter into details. Why? Because we try to hit 2 different targets with just 1 molecule. And here are some examples of the different series of inhibitors and activators which we have obtained. There are my colleagues, some of them who are here, Paola Gratteri, Alessandro Centino, Alessandro Bonardi, Andrea Mara, who are involved in the project. You may see the different series, which include monoamine oxidase B inhibitors, hybridized with a carbonic anhydrase inhibitor, cholinesterase inhibitor, carbonic anhydrase activator, and different other combination of these possibilities. All of them include series of 40, 50 different compounds, which were, of course, investigated and evaluated in different models. I will show you only two recent examples of, let's say, rather successful ones. The first one involves the hybridization of monoamine oxidase inhibitor of the coumarin type with a carbonic anhydrase inhibitor of the aryl sulfonamide type. With colleagues from our university, Gustavo Provenzi and others, we showed in the pharmacological models that they are helpful for protect against amyloid beta-induced neuronal toxicity, which is obviously a model of Alzheimer's disease. They also reduce oxidative stress and mitochondrial dysfunction, which are typical of this disease. I will not enter into too many details, but references here for who is interested. The second example, it's again a recently published paper just a few weeks ago, in which we observed enhanced recognition memory through the dual modulation of brain carbonic anhydrases with activators. You see the fragment contains, excuse me, the fragment contains carbonic anhydrase activators. I made an error. No. Okay. So we have inhibitors of the cholinesterases of the tacrin type and activators of carbonic anhydrase of different types. And also in this case, we have nice results in an animal model of cognition. Obviously, the compounds have been tested as activators of several brain-associated carbonic anhydrases of cholinesterase and butyryl cholinesterases, and we have nice expected results of the activation and inhibition. I was mentioning the pharmacological model, again done by Gustavo Provenzi. This is a social recognition model in mice, and again we have pro-cognitive significant effects when the mice were treated with these compounds, which were better than the combination of the two different drugs, of the drugs taken separately, obviously. I wish to thank my collaborators and the financing from this project, and thank you very much for your attention.
Thank you very much. Very interesting. Do we have a question? Okay. If not, we are a bit late, so we can proceed. Thank you very much again. So the next speaker will be Mauro Pineschi, and he will speak about development of innovative and affordable medicines for the treatment of obesity, diabetes, and neurodegenerative disease. He is from the University of Pisa. Please.
Okay, good morning to everyone. I'm very pleased to be here to present our invention related to the development of innovative, affordable medicine for the treatment of obesity, diabetes, and neurodegenerative diseases. We made part of this discovery under design of Tuscany health ecosystem, in particular this book 2, and the drug design part of this book. And in this framework, we wanted to realize a new small orally active drug, new small molecules being orally active to treat diabetes and neurodegenerative diseases that are often associated with diabetes. I use this term diabetes because it is amply known and widely accepted that there is a strict association between type 2 diabetes and obesity, pathologies that are often correlated. And in this speech, I wish to communicate to this audience about the possible introduction for the treatment of diabetes of new orally active small molecules, basically having three major goals that are shown here. So the individuation, the exploitation of a new mechanism of action, Also, an important factor is the reduction of the cost of the medical treatment, otherwise it would be not sustainable for the National Health Service. And the third is an increase in patient compliance by the use of an oral administration. So obesity is a very complex metabolic disease that affects up to 20% of adults worldwide, and it's a very serious factor that affects our health, predisposing our body to several pathologies. So there is not magic pill to cure obesity. However, there are new kind of medicine that are absolute game changers. You have certainly heard about this kind of medicine, this kind of drug, particularly semaglutide and tirzepatide, that are very effective for treating first diabetes and also obesity. These drugs are peptides, very effective peptides, are very famous, but they present also several drawbacks. For example, the very high cost of this, the medical treatment. The administration should be only by injection, normally, I mean, and also gastrointestinal side effects are very important, in particular nausea and vomiting. Also, a remarkable substantial muscle loss has been observed recently. Moreover, these compounds, being peptides, are not suitable for the treatment of neurodegenerative disease because are not easily permeant to the BBB. So, GLP-1 is an incretin hormone naturally secreted by our body, our gut, I would say, after a meal, and it is known to have several clinical benefits, such as the reduction of glycemic control, so the control of the glycemic, and also neuroprotection and cardioprotection are important effects. Other important effects, especially for the weight management, are the increase in energy expenditure and reduction of appetite. We came into this field in 2018 with a patent disclosing a completely new chemical entity that seems to have the right physicochemical feature to become an oral drug candidate to fight diabetes and obesity. And in general, as said before, the small molecules are easier and cheaper to produce with respect to peptides, and in particular, our compounds can be made in only 4 linear steps for commercial precursor. So are relatively easy to prepare, and the synthesis is scalable. So this is a very important point. Another differentiation of our compounds from existing solution is the fact that these compounds are not GLP-1 receptor agonists, but they act with a different mechanism of action. Just a remark, there is not to date a small molecule drug acting on GLP-1 pathway in the market, and there is a stiff competition for their development. So just having secured the chemical scaffold, you can see the marquee here. we made a so-called lead-to-lead optimization, finding lead compounds. You can see the huge difference of activity with respect to the hit previously found. And also in this case, we made other patents securing the intellectual property of our discovery. Just to make to show you the main features of these compounds, we have some proof of concept in vitro about the GLP-1 insulin secretion. You can see the increase in GLP-1 secretion on SCT1 cells, an increase in insulin secretion in INS1 cells, also human cells. This is importantly glucose dependent, the secretion of insulin. We have also an increase of cell viability and mitochondrial activity. This is also a very important point. We have also observed the insulin-like effects on this particular kind of cell. They are of neuroblastoma, human neuroblastoma cell line. And we have also no toxicity on hepatic cell line. About the neuroprotection, anti-inflammatory activity, we have a remarkable neuroprotective action of a neuroblastoma cell line with a substantial increase in two major cell survival pathways such as PAKT and MAPK, and a decrease in caspase 3 that on the other end is involved in neuronal death and apoptosis. And importantly, these effects are superior to liraglutide, that is one of the peptides commercially available that are permeant to the BBB. We have also marked a reduction of the pro-inflammatory interleukin 6 and an increase of anti-inflammatory interleukin 10. Just to show you some experimental details about this, you can see here the prevention of inflammation using several stimuli on human microglial cell line. You can see the effects of our lead compounds. And human microglial cell line are very important because they're the first line of immune defense in our central nervous system. Moreover, in addition to this, they are also involved in other cerebral processes, such as the modulation of a synaptic architecture and also in neurogenesis. So these are very important cells. We have also some proof in vitro on this cell showing the reduction of inflammation induced by amyloid beta proteins that are very important for the Alzheimer development. These proteins are known to accumulate to form amyloid plaques and to cause neurodegeneration and neuroinflammation, as shown here. And we have also optimal in silico drug-like properties. The compounds are permeant to BBB, a gastrointestinal membrane. We have no cytochrome inhibition and no genotoxicity. Clearly, we have also to demonstrate this experimentally. It's not just enough to make this in silico. We have also recently some in vivo data about an increased GLP-1 and insulin secretion, glucose-dependent insulin secretion on diabetic mice. And just the last result, because we have also interesting result in chronico with in diabetic mice, giving the drug orally, clearly, because this is a very important point. We observed a significant weight reduction compared to the untreated group, to the positive control, where we used metformin. Okay, so we have a consistent reduction of the weight, and also a significant reduction of triglyceride and glycated hemoglobin. No apparent toxicity was found. So at the end, so just to sum up this talk, now we would like to enter to the clinical trial, but you know that we need a lot of preclinical data to acquire, so we need a complete preclinical package, and to do that we are open to collaboration or to out-licensing our intellectual properties. At the end, I would like to thank all the people of the University of Pisa involved in this project. And clearly the financial support of Spoke 2 of the Tuscany health ecosystem is acknowledged. I also would like to acknowledge the University of Pisa for financial support derived from Dimostratori Tecnologici 2022. And these are my contact details. I would like to also to thank you for your kind attention. Thank you. If you have questions, don't hesitate to ask me.
Thank you. Just one quick question. Do we have? Yeah.
Yes. So thanks, Mauro, for the presentation. I have just a curiosity. I don't know if I skipped this part, but how did you arrive to the discovery of these molecules? Did you arrive to this by chance or by using virtual screening or what other methodologies?
Thank you for the question, Elena, because this is a normal question that everybody make to me. Because actually I am an organic chemist, and I found just this kind of scaffold just for casualty, I mean, for serendipity, because just sending these kind of molecules, I mean the heat compounds to an open innovation platform that was made by Eli Lilly in 8 years ago around. And just by phenotype screening, we discovered the activity. We have not target in mind, but just have this lucky event. Okay, thank you for the question.
Okay, thank you again. I think we can go and proceed to the next speaker, who will be Antonino Cattaneo from the Scuola Normale Superiore Pisa, and he will speak about intracellular nanobodies, a new generation of drugs. Please.
Thank you. Good morning to everyone. Scuola Normale is coordinator of Spoke 8 in the project, and I will be talking to you today about work that we are doing in my own group. So, scusa. Okay. So, let me define what is a nanobody. So, on the left there is an IgG that has variable regions and constant regions, and these are the standard way how therapeutic antibodies are used. Nanobody is a smaller portion of the antibody that encompasses only the two variable regions in the form of a single chain Fv fragment, or VHH, only heavy chain antibody. So my definition of nanobodies will be that either a VHVL, heavy and light chain, or a VHOVL single domain antibodies. And what I'll be talking to you about is how we have pioneered the technology over the many, many years to use antibodies as genes. Normally, therapeutic antibodies are used as proteins, and the therapeutic antibody market is the largest market of biopharmaceuticals, as has been mentioned before. And however, therapeutic antibodies by their format are limited to the world of extracellular targets, because you cannot have an antibody to access the intracellular portion of a cell in any tissue. And the intracellular environment is a huge source of disease targets. So our idea over the years has been, can we harness the power of antibody repertoires to hit intracellular targets? And so this is the main summary of work that we have been doing for many, many years. And the example that I will give you today is in the application of this approach in Alzheimer's disease. And I thank my previous speakers, to have made the introductions to the. So currently, Alzheimer's disease has a main extracellular view. So this is a dominating paradigm of Alzheimer's disease, a prion-like transcellular spreading of misfolded proteins, whereby vulnerable neurons start making a misfolded protein. This then is targeted, is exported outside the extracellular environment, and it hits neighboring cells. So like in a fire hitting a forest, the fire propagates and the neurodegeneration propagates. And the idea of therapeutic antibodies, as in the classical format, as are used in Alzheimer's, being developed in the field for the past 25 years for Alzheimer's disease, is to intercept the misfolded protein in the extracellular environment. However, we know that the efficacy is rather limited, unfortunately, for these therapeutic antibodies, and also there are many side effects. So this is really the problem in the use of therapeutic antibodies in Alzheimer's disease. And so this is. There is also another point, that this is a cartoon that you will find in any article on amyloid, and the cleavage of amyloid beta peptide is depicted as happening directly in the extracellular environment. This may well happen from the membrane APP, but as we have demonstrated, and as I will show you, we have demonstrated, and others have demonstrated, that the first production of A-beta occurs inside the neuron. So this is where really you want to hit the cell autonomous phase of neurodegeneration. And even more so, tau. Tau is an intracellular protein. It first misfolds intracellularly, and then it is, when the neuron degenerates, it is found extracellularly. So we really want to hit misfolded proteins inside the neuron. And so we want to hit the first cell autonomous damage to neurons where it is first done. And how to do this? And so we have developed this concept first, and then we have developed libraries, a platform of intracellular antibodies, intrabodies as we call them. They are intracellular nanobodies. And we can harness the power of the fact that we can derive from our libraries conformational, selective, and post-translational specific nanobodies against the misfolded protein. So this is what I will briefly tell you today. Now, and so the idea is that you have a gene that encodes for a specific nanobody, and you can target it to different intracellular compartments, and cytoplasm, nucleus, mitochondria, ER, membrane, synapse. And so that is really the power of how you can achieve subcellular precision coupled to molecular precision that the nanobody provides. And so this is the whole idea of the platform that we have been developing. And of course, the gene-delivered antibody is made by three moieties. The first moiety is the intrabody itself, so it has a specific binding to an intracellular target that can be post-translational modification specific or conformational specific. Then you equip this with a subcellular targeting sequence that will direct the gene after it is translated to a specific compartment so that you can really be subcellularly precise. And then one approach, you can equip the intrabody with an effector domain, for instance, a degron domain that will target the intrabody for targeted degradation of the recognized antigen. And so this is the idea, targeted degradation of misfolded protein inside neurons with molecularly precise nanobodies. And so we have libraries that we have developed that from which we call these libraries, splint libraries, single pot library of intracellular antibodies, whereby we can select by intracellular antibody capture technology a nanobody directly from the gene sequence that encodes for the antigen. So in our libraries, unlike any other technology that is available for making human antibodies, our technology, you have, I don't have the time to give you the detail, but the idea is that you have your target, your antigen is a cDNA that is encoded by a yeast cell, and then you have your antibody library, and the system is such that if an antibody in your library, or a nanobody in your library actually, binds to the antigen that all cells in your library express, this cell will survive, whereas the other cells will not survive. So you set up the system, you go home, 2 weeks later you come, and surviving cells will have the antibody that binds your antigen. Now, this is making it easy, but this is exactly from cDNA to antibody. So this accelerates enormously the process of antibody generation. And so these are our libraries, and we have libraries from naive libraries, so these are general libraries. We have them from camelids, from mice, from humans. We have patient-derived libraries, and so we can provide also patient-specific antibody libraries. And so we can go from DNA to nanobodies, no need to immunize animals, no need to produce and purify the antigen. We can do affinity maturation if necessary, and this is fast and scalable. So this is the first asset that our future startup company is going to build on. And second point is we have made a variant of these libraries to derive antibodies against post-translationally modified proteins. So these are, on one hand, a very difficult target because having a drug, no FDA-approved drug targets the post-translationally modified version of a target. On the other hand, they can be very, very disease relevant, and they are an enormous source of targets. So how can we derive an antibody against a post-translationally modified protein in the native conformation? We made we developed this platform that is called PISA, post-translational intracellular silencing antibodies. We have a granted patent on this technology. And so PISA platform is a discovery engine. We have proprietary libraries that can hit post-translationally or conformational proteins in a native conformation. We can translate them inside neurons or cells and target them to different compartments. And then these now are optimized for functional interference, for instance, targeted degradation. And what we are doing this now is in our Square Normale spin-off company, we will leverage the discovery platform for a new generation of drugs that are these nanobodies against the target proteins that are conformational specific, such as the beta oligomers, or post-translationally modified proteins, such as, for instance, phospho-tau. I will give you rapidly 2 examples. And so the drug candidates derived from the platform will be developed up to the end of regulatory preclinical studies, then to be licensed out. And so remind that I told you, I hope I convinced you that the first cell autonomous damage to neuron is done intracellularly. So both A-beta and tau are first formed intracellularly, they aggregate intracellularly, and then they are exported outside. And so the first product that we are developing in the lab is a nanobody against phosphotau. You know that when tau is abnormally phosphorylated, it detaches from microtubules, it aggregates in the cytoplasm, and this is where tangles are formed. So instead of targeting whole tau, that is a physiological normal protein, we want to target the phosphorylated version of tau and target specific phosphotau, but not normal tau, to degradation. This is the first, we have an active program in the lab working on this. And the second approach is to target A-beta oligomers, specifically that we have developed by Giovanni Meli and my group over already 10 or 12 years ago. And these nanobodies specific for A-beta oligomers, we have demonstrated that both in cells and in vivo in mice, we can use them, first of all, to demonstrate formally that when we express this oligomer-specific intrabody in the endoplasmic reticulum of a neuron, there the oligomers are first formed, intracellular oligomers, despite any cartoon that you will see in any article. And so the first site where oligomers are formed is in the endoplasmic reticulum, and by expressing a nanobody in the endoplasmic reticulum, we can target them, we can block them there and avoid secretion in the extracellular environment. So we can block the fire of the forest by protecting the neurons inside, and we can show that this is effective in mice models in vivo. So the idea now is that as a milestone of our project in the PNR, in the ecosystem, we are setting up a startup company based on our granted patented technology, based on 25 years of having pioneered this idea of using antibodies as genes. And we believe that this technology can open the way to the world of conformational specific misfolded proteins inside cells, and for what we call a subcellular precise pharmacology. And so, of course, we have an expertise on neurodegeneration. This is where we shall develop this technology, but the value of the technology platform is very well beyond the internal therapeutic focus on central nervous system. So let me thank you for your attention, and my intrabody group at Scuola Normale, Gabriele Ugolini, Simonetta Lisi, who is really the queen of the libraries, and then PhD students Angela, Rachel Bitonti, and Martina Dassolo, and Rajesh Jacob. Thank you very much indeed.
Thank you very much for this fascinating presentation, and we already have a question there.
Amazing presentation, thank you very much. Is it possible to think of nanobody drug conjugates? Did you think of this?
Yes, well, that's a very, very good point. Of course, once you have the core of the nanobody with an exquisite molecular specificity that you can engineer, select, or tailor, and then, of course, we can add either effector functions if you deliver these as genes, as I try to convince you, but also if you deliver them as protein, you can chemically couple with chemical conjugates of interest. So this is definitely around the core of a nanobody that we can derive from our libraries. We are very, very happy to sit down and think about possible conjugates of interest. That is the short answer. Thank you, Claudio, for.
Have another question there.
And nice presentation, thank you. What about the stability of this nanobody inside the cell? Because this is a very important point. When single chain FEV are delivered through the blood, they have a very short half-life. In this case, something is changed.
Yes, thank you very much for asking again. This is why we developed intracellular libraries and biological selection. We, around the universe of all possible antibodies in nature, our selection system is selecting a nanobody under conditions of intracellular expression, so we are certain that the nanobody that we select have been selected under conditions of intracellular expression. So we are sure that they are stable enough to work because we have selected a priori those nanobodies under those conditions. That is exactly why we don't use phage display libraries or ribosome display libraries or other libraries or even monoclonal antibody from humans, because we select intracellular nanobodies, our SPRINT libraries. Thank you.
Thank you. So if we don't have any further question, we can go to the next presentation, will be by Anna Rosa Arcangeli from the University of Florence. She will speak about novel immunotherapies in cancer, targeting beta-1 integrin complex through recombinant antibodies in combination with cholesterol-lowering drugs. Please.
So I think it's a very long title, this, but we remain in the field of immunotherapy and antibodies. So let's see. Okay, cancer immunotherapy. Cancer immunotherapies comprises cell therapies and antibodies. We will focus on antibodies, and in particular on specific antibody formats. Professor Cattaneo already introduced you to the nanobodies, and we will go deeper in this field. So when we talk about antibodies, we have to talk about antigens. So in our case, we have an antigen, of course, and a recombinant antibody. Which is the antigen? The antigen is a complex, so not just a protein, but a complex between two proteins expressed on the plasma membrane, selectively expressed on the plasma membrane of cancer cells, and the two proteins are represented by an ion channel and a potassium channel, to be precise, hERG. hERG is physiologically expressed in the heart, so many of you know hERG. But the other part of the antigen is represented by an integrin, by an adhesion receptor, and in particular by the beta-1 subunit of adhesion receptors of integrins. As stated before, so this complex is present only in cancer cells as a complex, tightly bound proteins. The antibody is a single chain bispecific antibody in the format of a diabody. So we started with two monoclonal antibodies, one against hERG and one against the integrin, the beta-1 integrin. Then we produced the CFVs, and then we joined the two. We worked a lot on the linker between the two arms of the antibody to have a true diabody capable of binding only the complex and not the two proteins when they are expressed separately, not as a complex. We published the results of this development. Before publishing, we patent the idea and the product, and the patent is owned by the University of Florence, but was licensed some years ago to MCK Therapeutics, which is a spin-off of the University of Florence, who was in charge of extending the patent, which is now granted in Australia, Canada, China, and so forth. So it is fully protected. And now we come to the title of this presentation and to the work we did on this topic, thanks to the financing from the THE, and in particular we belong to the SPOK2 group. And the title states that we can perform immunotherapy in cancer combining the antibody, the diabody I mentioned, with cholesterol-lowering drugs. Why? Which is the rationale of this? The rationale is the fact that the complex, which I mentioned, is selectively present in cholesterol-rich plasma membrane microdomain. So what which we usually call lipid rafts, and which are the triton insoluble part of the plasma membrane. In this specific location, the complex triggers an intracellular signaling pathway which involves classical molecules like PI3 kinase, AKT, RAC1, and so forth, and at the end, it controls cell cycle progression through cyclins and in motility and invasion through the remodeling of the actin cytoskeleton. And so cholesterol-lowering drugs, statins, what do they do in these cells? I'm referring to cancer cells, of course, and in particular to pancreatic cancer cells from here onward. So statins can lower cholesterol in the cells, can dissociate the complex, which is in lipid rafts, and similarly to what exerted by the diabody, by the single chain diabody. And if we combine the two, the complex is no more in lipid rafts, is no more present in the cells. And the combination of statins with the antibody, which I called here SCDB, HER1, beta-1, let me say just SCDB, the combination of statin with SCDB lowers the proliferation rate and the invasiveness of pancreatic cancer cells. What happens in vivo? In vivo, this is the pancreatic cancer model, an orthotopic xenograft obtained by ultrasound guided injection of human pancreatic cancer cells into the pancreas of mice. You can see here also the images of the ecography, and the combination of statins with the SCDB greatly lowers the growth of the tumor and improves the survival. So in the last 2 or 3 slides I showed you, there was this new patent, because we have applied, submitted with the University of Florence, thanks to the THE grant, a new patent which has been submitted as PCT and which covers all what I showed you today. And even protected by the patent are these results that are, quite interesting for us. So we show that in the cells, in the pancreatic cancer cells, that the presence of the hERG, of the complex on the plasma membrane, affects the capability of the combination to, in this case, to kill the cells. This is one point. The second point that I think is very interesting is the fact that the combination of statins with the SCDB works even more and is synergistic with low doses of chemotherapeutic drugs, oxaliplatin. So this combination can be proposed to overcome resistance or to be combined since the beginning of treatment with chemotherapeutics like oxaliplatin that for example, in pancreatic cancer is employed as a neoadjuvant, so before surgery. And so we are close to the patients now. What happens in patients, which is the scenario? Here again, pancreatic cancer. The complex is expressed on the tumors in I would say, a high percentage of cases. But what is relevant for us is that it represents a negative prognostic factor. So the survival is worse in patients whose tumor has the complex on the cells, on the tumor cells, and this occurs either in the total population or in some population which are treated with statins for comorbidities. The therapy with statins is very common. So we believe that, okay, we can use statins alone or in combination with the SCDB or other chemotherapeutic drugs for the treatment of patients with pancreatic cancer, especially those who express in their tumors the complex HERG beta-1, because it is a negative prognostic factor, and because the presence of the complex of the channel makes the combination therapy to be effective, or supposedly makes it to be effective, and this is the title of the patent application which I mentioned before. And last but not least, in the previous slide, I highlighted in red the fact that the combination could be applied not only to pancreatic cancer, but also to other cancers. And indeed, starting from these studies, starting from this patent, what I call a cascade call grant was granted from the University of Florence. The name of the project is REPSTAT, so it's easy to understand what does it mean. So repurposing statins, and in this case, two companies, GA Lab and AWA Medical, will work together, and will work together also with MCK Therapeutics, because the antibody is produced by MCK Therapeutics, to test the combination in breast cancer, because the rationale, again, is the fact that the HERG beta-1 integrin complex is highly expressed in triple-negative breast cancer. So, thanks to the European community, to the Tuscany, to the university for granting these studies, but the thanks are to Claudia, who is present in the audience, and Jessica, my collaborators and co-inventors of the patent. And thanks to you all for listening to me.
Thank you very much for this very interesting presentation. We have a question here.
Thank you, Anna Rosa. Fantastic talk as usually. I have an idea. You probably know that we have a compound discovered in Florence in phase 1b/2 clinical trials exactly for pancreatic and triple negative, which is a sulfonamide carbonic anhydrase 9 inhibitor. I think your approach with targeting HERG1 statins would benefit from a combination with this type of carbonic anhydrase 9 inhibitor, which is obviously, very well important in hypoxic tumors.
Yes, it's fantastic, and this idea, I would say this was an idea that we already pursued with Claudia, and we worked with you. I don't know whether it is the same compound, yeah? And Claudia was able with your group to do an ADC, but we applied to kidney cancers. Maybe the possibility of using in pancreatic cancer and triple negative is fantastic. Thank you, Claudio. Hi.
Thank you again.
Thank you. So welcome on stage Elena Petrici, next speaker. She'll be talking about ADC. Actually, I learned a new acronym today. It's antibody-drug conjugates redefining precision medicine for cancer and beyond.
Thanks. Thanks, Andrea, for the presentation, and thanks to the organizing committee to give me the possibility to present you our efforts in developing new antibody-drug conjugates for cancer and beyond. Antibody-drug conjugates, as many speakers has shows this morning, are composed by a monoclonal antibody specific for antigen, hopefully selectively expressed in cancer cells, and connected by a properly designed linker to a cytotoxic molecule here called payload. When the ADC arrives in tumor cells, it's able to bind its specific antigens, and usually the mechanism of action is an internalization of the ADCs by endocytosis, and the endosome then matures into a lysosome. Here, inside the lysosome, the ADC can find different pH conditions and enzymes that are able, depending on how we design this linker, to release the drug inside the tumor cell and just producing cell death. There are available even antibodies that are not internalized and that can be used to produce ADCs. Example from Philogen and Philochem are reported in the literature. The difference is just in the linker, on how you design the linkers to get the drug be released outside the cell. Here there are the ADCs actually approved in the market. They are 14, and they start to be approved in 2011, the first one, and actually there are more than 100 ADCs in clinical trials. Why companies are actually so interested in ADCs? We start to work on this field in 2014, so we were almost here, just 2 ADCs were approved, and the idea was that if you have a molecule that is already in the market for the treatment of any disease, and if you have a monoclonal antibody which is actually applied in immunotherapy, you have just to connect them together, and you can skip most of the clinical trial phases, so you can arrive in the market quite easily. Actually, the situation is a little bit different, because going on with the discoveries of new ADCs and with the technology, it's much more mature, so there are many requirements reported, but generally it's a kind of Lego approach, and it's even easy to have something that you can patent, because you have just to change the monoclonal antibody or the payload or the linker, and you have something new. If you have a linker payload system and you change the monoclonal antibody, you can have something which is active against different targets, and that you can use in different diseases. And you can collect a library of different ADCs for different applications, and in this game there is a lot to do, working both in new payloads, new linkers, new monoclonal antibodies, and even in new way to connect the linker payload system to the monoclonal antibody. Any part of this explains a crucial rule. Our idea is that this approach will be interesting in the future in precision medicine, because we can think to have a patient coming in the hospital and you have the diagnosis of a cancer, so you can go on with the analysis and you can establish that his or her tumor cells express a specific antigen. So you can try to synthesize a monoclonal antibody specific for this antigen. In this assay, you can find small molecules active on these cancer strains, and you can put them together and treat the cancer of Mary or whatever, and have hopefully a complete recovery. I say a complete recovery because in our experience, what we did with the ADCs we develop is that we have a complete eradication of cancer. We work on melanoma, in non-small lung cancer, in pancreatic cancer strains in different way. With some of our molecules, we are actually in monkeys. And what the biologists observe, I'm an organic chemist, so this is not my part, but is that we have a complete eradication of the cancer. And working on this part, in the chemical part of ADC development, what I realized is that, okay, it's a Lego approach, but working on that, it's much more similar to a jigsaw puzzle, in which do you not really need only to have the right pieces, but you even need to put them together in the right sequence. Otherwise, you are not able to have your final compound, because it's quite common in our lab, I don't know in the others, to work for months in the synthesis of a linker payload system, and then arriving at the bioconjugation step, we are not able to connect the monoclonal antibody with this system. So you have to synthesize a new linker or to change the amino acid residues of the monoclonal antibody you want to use, or you have to pre-functionalize the monoclonal antibody, but in any case you lose a lot of time. So that's why in my research, in the context of the Tuscan Health Ecosystem Project in Spoke 6, I start on study and developing a machine learning model for the prediction of bioconjugation. And we have been able with Lorenzo in our group, starting from the data that we have in our lab, because working on this field since 2014, we have a lot of failure in bioconjugation, several success, but we have data that were homogeneous, and this is crucial to have a good machine learning model. And our system, it's able actually to predict if a linker payload system can be bioconjugated or not with your monoclonal antibody, and eventually how many molecules of your linker payload system in certain experimental conditions can be charged over the monoclonal antibody. This is a crucial point, because the drug to antibody ratio, that means the number of molecules that are charged over the monoclonal antibody, it's a very important parameter to obtain an effective ADC. And you need to be in between 2 and 8 to avoid a loss of efficacy of the monoclonal antibody in recognize the antigen or aggregation processes. So at the moment, this is under reviewing process, and we obtain good results in prediction. Any part of the ADC, it's important. So another part of our research, it's focused on development of different linkers. Here I just reported a simple system, but we developed several over the year, and we are still working on this part. And we work even in finding new molecules, new functional groups that can be used for the bioconjugation with different amino acids over the monoclonal antibody. Even this part, it's important, because these two together are the ones that are important to get for the bioconjugation an homogeneous ADC, and on the other hand, a linker that need to be stable in the plasma, but need to be able to release your drug once the ADC is internalized. Then we move to other applications than cancer by changing the monoclonal antibodies used, and particularly in collaboration with Toscana Life Sciences and the group of Claudia Sala, we work on anti-COVID ADCs, as well in aptamers drug conjugates and in nanobodies drug conjugates in collaboration with the Institut Pasteur, again for application in virus. But this is possible just because several years ago, when we start, we have been able to develop, to demonstrate that it's not mandatory to have a cytotoxic molecule to have an active ADC. This is a crucial point, because when we start, and even now, if you look at the ADCs approved in the market, I reported in the same colors the ADCs that are composed by the same payload, that means the same cytotoxic drug and just a different monoclonal antibody. And except for the green one that have a topoisomerase 1 inhibitor as a payload, all the others are charged with natural products which are very, very cytotoxic. The EC50, it's in the range of in the picomolar range, and the idea at the beginning was that if you have a monoclonal antibody and you have just 2, 3, or 4 molecules that are connected on it, to have something which is active in tumor cells, you need something which is very, very cytotoxic. And this was the first choice. And when we start, the idea was that it was not possible to use other type of molecules to have this activity. And using cytotoxic molecule, it's somehow limiting the application of ADCs to cancer. And on the other hand, if by chance you have the release of the drug in the plasma or in other tissues, you have something which is really cytotoxic, and you have a high risk. So the question was, in collaboration with Alpha Wasserman, if it was really mandatory to have these kind of molecules charged over a monoclonal antibody to have an effective ADCs. And the idea, as the company developed in collaboration with our group an inhibitor of histone deacetylase, was to investigate if it was possible to have an epigenetic modulation by using ADCs. And it was possible. We have been happy for this. Since we discovered and we developed this part by using vorinostat and then all the HDAC inhibitors reported in the literature, again with still with Alpha Wasserman, we realized that the hardest part when you are using compounds that are not very cytotoxic, it's represented by the biological assay you have to do. Because at the beginning we have no data in vitro assays for any of our compounds. And this is the demonstration on how a collaboration with a company can be an added value in this case, because if it was an academic project, we probably stop saying, okay, it's not possible to do that. But the company was convinced that we can get the activity, and so we directly move to the mouse model. And in vivo studies, the products, some of the products were really, really active. Then we went back and we set up a system for the in vitro assays. But in vitro assays, usually for the first time, you do them in different models, completely fail. So we learn something from this. And then we move to different payloads, such as MRE11 inhibitors. We developed some years ago. These ones are good for to be used as radiosensitizers, so we are bioconjugating them. And then we have small inhibitors, that means modulators of the Hedgehog signaling pathway. And we did this even thanks to the ANAIRC project, and as well as proteids. So the ability, the demonstration that it's possible to use even molecules that are not cytotoxic open to new projects. Here they are reported, some we are working on antiviral therapy, antitubercular one, as well in the use with using our linkers, especially our payloads, to use different biomolecules as carrier for our compounds, in particularly aptamers for the glioblastoma, and ferritin inside the context of spoke number 6 with Professor Castagna in the University of Siena for the treatment of anaplastic carcinoma of thyroides. This is possible thanks to the work of a lot of people. I didn't report the biologists. They are reported in the patent and in the papers. I reported the people in my lab actually working in the project. Juicy is here with us, and basically she's coordinating most of the part. She came back thanks to the project from a company to work in the university, and she brings with her an added value for my lab. So I'm really happy, and I really thanks this project even for this part. Giovanni, he's has a grant from the project and is working in the cyclopamine part, while Demetra is working in developing new linker, and Lorenzo in the machine learning approach. Maria, it's more focused on the payloads, and Francesca, it's actually working in phylochem, and she basically works in different parts, such as cyclopamine and linker, and I would like to thank her as well. And for the financial support, obviously, I thank the Spoke 6 of the Tuscany Health Ecosystem and the Regione Toscana for these different projects. PRIN 2022 for the part of anti-retub on tuberculosis, PRIN 2020 for the FAITH project on COVID-19, AIRC and Alpha Wasserman and Ricordati for the ADC's projects in cancer. And you're all for kind attention.
Thank you very much, Elena. Thank you so much. Any questions from the audience for Elena?
Thank you very much for your talk. I had a question on how much, if it is a problem when in an ADC complex, the escape from the endocytic compartment where the complex is internalized to progress onto the desired target, so to escape the endocytic bicycle.
You mean the bystander effects?
No, how precise you can be in once the payload is internalized.
Okay, so once the payload is internalized, usually you are quite precise. The bystander effect, the so-called bystander effect, it's quite known. So in some cases, what happens is that the molecules, once released inside the cells, it's moving outside the cells and in the surroundings, and it can act in the cell that surround the target cell. This is not usually considered as a problem, but it's considered as an added value, because you can in this way target molecules that do not express the antigen and are still tumor cells. It depends on how this part is important and how many of these molecules are able to pass the membrane. But in general, there are even some softwares and systems developed over the years that are able to predict, depending on the molecule, on your payload, how many of these molecules will move outside the cells and enter in the environment. Thank you.
I invite my co-chair to move to the stage and to talk about, Luisa Bracci, of course, to talk about branched peptides for therapy.
What is that? Okay. Okay, I'll be very quickly try to explain why branched peptide, but the story is really too long to be explained into detail. So now, many years ago, we discovered that when peptides are synthesized in a branched form, which can be easily obtained by chemical synthesis, using a core made by lysine amino acid, which is schematically reported here. When these peptides are synthesized in this form, they become extremely resistant to proteases, and particularly to peptidases, which means that they acquire features that can be useful for in vivo use. So you see here that when we discovered that, we use this technique and we move to monomeric to branched peptide, and for several applications which you can see here. Oh, sorry. Okay, so in this presentation I will show something which is regarding the application of branched peptide in cancer, and in a very new project which we started thanks to the Tuscany ecosystem regarding possible bone regeneration. Then in the next presentation you will hear about another branched peptide which was used for years for treating bacterial infection. So, regarding the target, the cancer targeting by branched peptide, many years ago, we synthesized a sequence which was practically derived from a natural peptide, but the story is really too long. And because we realized that when this peptide was synthesized in a branched form, note, only when it was synthesized in a branched form, not in a monomeric form, it was extremely specific for recognizing many different cancer tissue. As you can see here, and in many different cancers, this was published now many years ago, colon, pancreas, urinary bladder, using the peptide for staining the tissue, this is what was obtained. So the cancer tissue was extremely, the staining of cancer tissue was extremely higher compared to the staining of the healthy tissue from the same patient. We collected many different tissues, and so we could analyze the statistical, we could perform a statistical analysis on what we could see in the microscope. And later on, we discovered which the target was, and that was interesting because the target of this sequence, branched sequence, were the glycosaminoglycan sulfated chain of heparan sulfate proteoglycans. And also thanks to our studies, it was demonstrated that many cancers, they express extremely highly sulfated heparan sulfate proteoglycan, much more than healthy tissue. So they overexpress these, but more than that, they oversulfate these heparan sulfate proteoglycan. So we used that, thanks to this cancer selectivity, we thought that it was possible to use that peptide for targeting cancer, and we, of course, we started with analyzing what we got in the cell for cytotoxicity, but then quickly we moved in vivo model, and this is a model where we use this branched peptide conjugated to paclitaxel. And we compare what we got with the name of this peptide is NT4, conjugated with paclitaxel, and this is what we got when we compared not only with the control group, but even with group treated with the same concentration, the same amount of paclitaxel. So the use of this peptide conjugated to paclitaxel in this orthotopic breast cancer model, mice model, was really very good, and in some of these mice we have clearance from the disease. But then we, knowing that the target was the heparan sulfate proteoglycan, we also wanted to use those peptides as a tool for analyzing the function of these molecules in cancer, which is not completely known. And so, since we had a tool which was specific for the heparan sulfated chain of these membrane proteoglycans, and those are known to have a role, not clear how much, not clear how autonomous, particularly in cancer cell adhesion and migration. And so we use the peptide, the branched peptide, now unconjugated, so with nothing conjugated to, for analyzing the effect on cancer cell migration. This is what we got with different cancer cells. This is the pancreas adenocarcinoma, and these are the very famous breast cancer, human breast cancer aggressive cell line. And by adding the peptide, it was clear that the migration was lowered. And then we also analyzed how this peptide was effective in another in vitro model for cancer invasiveness, which is the ability of the cell to make colonies in vivo. So you just seed the cell at very high dilution. You wait for some days, and and then you measure how many colonies are formed. And in the presence of the peptide, colonies in both the pancreas adenocarcinoma, and particularly in the breast and also in the breast cancer cell line, the number of colonies was much, much lower. And so we started to try to understand what these heparan sulfate proteoglycan were doing, and which was, and we are still analyzing, which the role is in cancer cell migration. And what we noticed is that by using the peptide, but also using an antibody which is commercially available, which recognizes heparan sulfate on this heparan sulfate proteoglycan, when you have migrating cell, those sulfated chain stay on the border. So they are much more expressed on the border of migrating cell and much less in the interior. And we also wanted to see which was the proteoglycan which was carrying those heparan sulfate chain, and those are different in different cell lines. And in this case, we had the Syndecan-4. I don't have time to explain what Syndecan-4 means, but Syndecan-4 is one of these heparan sulfate proteoglycan which can be directly involved in a signaling, because it has several intracellular domains which are known to be linked to different signaling. Not exactly how, but they are known to be related to the intracellular signaling for even, and particularly for the signaling for the organization of the actin cytoskeleton, which means migration. And okay, so this, I will very, very quickly move to the other, to a completely different topic, which is the new project that I told you we initiated thanks to this Tuscany Health Ecosystem program, so it is pretty new, and completely another thing. So we wanted to see whether it was possible to select branched peptide, which can then be synthesized in a branched form, which we usually do in our laboratory. And the target that we wanted the target of our choice was sclerostin. I don't have time to go into detail of the signaling, but sclerostin is an inhibitor of an important signaling, which is Wnt. And by inhibiting Wnt, sclerostin is one of the most important regulators of bone regeneration. So as long as it is there, the bone regeneration is off. And in order to increase the bone regeneration, you have to get rid of sclerostin. And so what we wanted to do was to select a peptide able to bind to sclerostin and eventually interfere with the role of sclerostin in the Wnt signaling regulation. So inhibiting sclerostin means to switch off the Wnt bone regenerating signaling. And for this reason, sclerostin is already considered as a very important target for possible therapy for osteoporosis. So I will very, very quickly say, because we are just at the beginning of this study, so what we did was what we did several times for our targeted peptide. So it was to select the peptide by phage display. We spent some time for setting up the methods which could enable then the possible analysis of the function of peptide that hopefully we could select. But since the beginning, we also performed two different panning, so the two different selection of these phages. One was the classical selection by elution buffer by pH, and the other one was the was a competitor panning because sclerostin is an heparin binding protein, and it binds to LRP receptor, and as many binders of the LRP receptor, they also bind to heparin. So they are heparin binding protein. So we also performed a selection by competition. So the phages bound to the antigen were detached by adding heparin. So the heparin could bind to the immobilized sclerostin, which was used for the panning, and hopefully we could select those phages that are that bind heparin, which means that hopefully they can more probably bind also LRP receptor. And I would like to tell the end of the story, but unfortunately I cannot, because we are still working on that. We already have some peptides. We synthesized in a branched form, and we are just in these days currently analyzing the function of this peptide by analyzing the effect on wind and signaling, and before that the binding and the affinity of this peptide. I think this was my last slide, so I don't have the photo of the collaborators, but many of them are here, so you can see them. And so I want to thank all the group and of course you for your attention. Thank you.
Thanks a lot, Luisa, for this nice presentation. Any questions? No, doesn't seem so. So let's, for the sake of time, let's move forward. I invite Alessandro Pini on stage. He'll be presenting functional devices for the treatment of sepsis.
Thank you, Andrea. My name is Alessandro Pini. Here you can see my affiliations. Oh, okay, just trying the pointer. My affiliation, I'm professor of biochemistry at the University of Siena, biologist at Ospedale Rescote in Siena, and founder, along with Professor Bracci and Falciani, of this small company, Setlands, and these three institutions are collaborating for the setup of new medical devices for the treatment of sepsis. So, I would like to start my presentation just with a short introduction about sepsis, which, you know, is a life-threatening, very severe life-threatening condition, which is triggered generally by a bacterial infection, bacteria which release toxins and pieces of bacteria which generate a sort of cytokine storm inside the body, together with leaky vessels, clots, and so a sort of inflammatory process out of control, which can generate organ failure and septic shock and death of patients. Okay, here you can see a sort of schematic representation of the evolution of sepsis until the septic shock. Anyway, the most important part of this slide is the numbers. It is calculated that about 27 million of people develop sepsis per year worldwide, and among these 8 million people dies for sepsis. And on the left you can see the commercial value of sepsis. It is not so beautiful to talk about the commercial value when regarding pathologies, but you know how important is the companies which are interested in setting up new treatments, new medicines, etc. for a pathology. And the sepsis market is expected to rise around $1 billion of dollars in the next 10 years. So the interest is very, very high for the treatment of this disease. Also because at the moment, in 2024, no specific therapeutic regimens, or better, medicines are present, exist for the sepsis treatment. Here you can see a schematic representation of a sepsis patient in an intensive care unit, which is initially cured with administration of antibiotics, of course. The sepsis is started with bacterial infection generally, and then many other supportive therapies like fluids, the administration of vasopressors, insulin therapy, and artificial ventilation of lungs, etc., etc. But it doesn't exist a molecular target therapy at the moment. So it is important to have something that can cure directly sepsis when it is diagnosed at the beginning, in an early stage, let's say. Actually, in the last 20 years, let's say, some different medical devices are used in the clinical practice for in extracorporeal circulation system for the purification of sepsis blood from toxins or cytokines. Here you can see the representation of the extracellular circulation system and some some examples of these already commercially available medical devices. The problem of these medical devices already on the market is that no one of these is able to remove lipopolysaccharide from gram-negative bacteria, or lipoteichoic acid from gram-positive ones, or cytokines at the same moment. And this is the main reason for which the use of these devices is not resolutive at all for the sepsis patients. So our project is based on the construction of new medical device which can do all these things all together. And the new medical device is based on this antimicrobial peptide called SET-M33, which was isolated in my lab many years ago, actually. And it was initially thought for different applications, for the setup of new antibiotics. Here you can see a selection of many articles that we had in the last years for this different application of this peptide, which is synthesized in the branched form Professor Bacci talked about before, very stable form, and whose mechanism of action is based on the presence of positive charge on the peptide sequence, which can react with negative charge of biological structure like LPS or LTA, or bacterial membrane just producing bacterial perturbation and destruction of the bacterial structure. So the idea is to use this peptide for conjugating it with a biocompatible resin, and the first prototype that we used was based on agarose beads. So we synthesized the peptide with functional groups. This tile group is used for the conjugate covalent bond with a iodoacetyl group of agarose beads in order to have a stable covalent bond, in order to have a final resin to be used in extracorporeal circulation. Of course, all the experiments that I am going to show you are in vitro experiments. We are not in the clinical evaluation. And different patents have been filed for the application, for the construction and application of this new device. The first prototype was used in vitro. Here you can see some very promising experiments for the removal of LPS from physiological solution or sera spiked with a known amount of LPS. You can see that the removal was higher than 85% with respect to the same cartridge without the peptide. Same thing for the removal of lipoteichoic acids from gram-positive bacteria, and more or less the same thing for living bacteria from the sera of spiked with a known amount of bacteria. So the cartridge worked very, very well, but we had a problem, a very technical and physical problem. The resin is very thin, and we had in the circulation system a sort of counter pressure due to the high packing of the resin. So it was not so suitable for a real application in patients. So we found a new collaborator. This company, Medica SPA, which is based in Italy, near Modena, a company which works with filter for water purification and biological fluids purification, which provided us a new matrix based on this molecule, polyether sulfone polyvinyl pyrrolidose, so PES for France, let's say, which can be produced in fibers or granules, and a small or large cartridge can be filled with this material, biocompatible materials already used in different applications. The problem was how it is possible to conjugate covalently the peptide to this material without functional groups. The idea was the use of the sterilization phase made by beta race ionizing race, which produce free radicals. So the incubation of the PES with the peptide in the same cartridge, then sterilized with beta race in order to produce free radicals which generate covalent bonds, a specific covalent bonds between the two compounds, in order to have, how you can see here, a stable matrix. Now I skip all the experimentation that we performed for the quantification of the amount of peptide covalent bound, but anyway we had a good quantification of peptide, so we pass to the efficacy of the system. Here you can see an in vitro system with a special machine for circulation using whole human blood or sera or water solution spiked with LPS or bacteria, sterilized with the system of beta race, and then circulate through the system with a known amount of LPS. And here you can see the kinetic of LPS removal. After 15-30 minutes, the removal of LPS is practically around 100% with respect to the cartridge not conjugated with peptide. So it is practically after the first minutes of circulation, the removal is very, very high. More or less the same thing for bacteria. In this case, it was used Bacillus diminuta, which is the reference bacterium used for this application. And even in this case, after the first 15 minutes, the specific peptide in the cartridge remove all the bacteria. Even if in this case, a non-specific entrapment of bacteria are present in the non-specific cartridge. This is the result of the last months, let's say. A very, very important aspect of this cartridge, which is able to remove also inflammatory mediators and cytokines. In this case, we can see the example of C-reactive protein. C-reactive protein, which is a very important biochemical parameter for inflammatory process, as you know, but it is also very, very important in its activity for the evolution of an inflammatory process, above all when it is out of control. And you can see how the cartridge is able to remove C-reactive protein around 100% with respect to the control cartridge. So the last slides just to evaluate how the sera of sepsis patient after the circulation in this cartridge and the removal of sepsis trigger or inflammatory mediators change. Here highlighted, you can see the amount of C-reactive protein in the serum of patients with sepsis, and after the circulation, you can see the very strong reduction of PCR. So this is not a reduction, this is an abatement practically. More or less the same thing for procalcitonin, which is another parameter used for the evaluation of inflammatory process in sepsis, which is a very important reduction. It doesn't work at all. Okay, even if in this case we have a specific reduction also in the control device. Next step, we are in touch with international companies for the large-scale peptide production in GMP condition in order to arrive to the animal models of extracorporeal filtration, blood filtration. Of course, it is impossible to do this with small animals because the extracorporeal circulation machine are not do not exist for mice or rats. So we have a project of experimentation with pigs and then to the clinical evaluation. Here you can see the people working on this project from the Setlands, the Hospital of Siena, Medica SPA, which provided the resin, and the University of Siena, where we work, and Giovanni Cappello here present in the audience, which is the most important researcher involved in this project. Thank you very much for your attention.
Thank you very much, Alessandro. Are any questions from the audience? No, I can't see any arm raised. Thank you, Alessandro. So let's move to the last speaker of today, this morning actually, first session. Giovanna Batoni from University of Pisa. She'll be presenting multi-approach strategies to prevent or treat infections in the era of multi-drug resistance.
Okay, good morning to everybody. I thank the organizers and especially Luisa for inviting me to speak here this morning and to present our more recent data. Antimicrobial resistance occurs when microorganisms no longer respond to antibiotics, rendering the antibiotic therapy ineffective. And actually, as it was mentioned this morning, antimicrobial resistance today represents a silent pandemic, estimated to have contributed to over 4.9 million deaths worldwide in 2019. And just to put this number into perspective, estimate of the number of deaths caused by the SARS-CoV-2 during the first year of the pandemic range between 1.8 and 3 millions. But not only the death, so the loss in human lives, but also the indirect effect of antimicrobial resistance are very dangerous, as key and widespread medical procedures such as intestinal surgery, cesarean section, joint replacement, or other could become too risky without the support of an antimicrobial therapy. So for this reason, it is believed that because of antimicrobial resistance, the achievement of the modern medicine is at risk. And if the trend is not quickly reversed, it is estimated that in 2050, death from multi-drug resistant infection will probably exceed those due to cancer. So it is clear that in this scenario there is an urgent need of antimicrobial strategies alternative to antibiotic use that may target antimicrobial resistant pathogens. And many of these strategies are today under investigation in many laboratories all over the world. And I should say that the task is not easy at all, because these antibiotic alternatives should be highly target specific, non-toxic, not inducing resistance, and possibly also economic and environmental friendly. So in this context is working the group of the researchers within the Spoke 7 of the sub-project 5 at the University of Pisa in the Tuscany health ecosystem, and some of them are working on some of these new strategies, such as phytochemicals, antivirulent strategies, drug repurposing, metals, or phage therapy. Of course, due to time limit, I do not have time to talk about all of them, but I will concentrate on the possible use of probiotics and postbiotics to prevent or treat infectious diseases. The local application of harmless microbes, the so-called probiotics, is an emerging antimicrobial strategy called bacteriotherapy. And the general strategy is to exploit the natural competition among bacteria, using beneficial bacteria to fight against pathogenic ones. And the mechanisms of action are several. For example, probiotics may interfere with the pathogen adhesion to epithelial layers of the host. They may produce an array of bioactive substances that contrast pathogen growth and virulence, and also they may stimulate the immune system, reduce inflammation, enhance mucosal barrier integrity, and also stimulate healing. But we have to consider that behind viable bacteria, also probiotic-derived inanimate preparations that are actually called postbiotics are an emerging antibacterial strategies, and they may have some advantages in terms of safety, especially in vulnerable or immunocompromised individuals, and they might be easier to produce and storage. So in my presentation, I will show you two major examples of our work. The first one, the use of live probiotics for the treatment of infected wounds, and the second one, the use of postbiotics for the treatment of respiratory infections. Chronic wounds such as diabetic venous pressure ulcers affect millions of people worldwide, and the incidence is expected to rise with the general aging of the world population. So a few clinical studies have demonstrated that the local administration of probiotic to an infected wound may have positive outcomes in terms of reduction of the bacterial load and also wound healing. But the problem is how to administer the probiotic to the infected wound. So the challenge is to develop adequate delivery systems of a live probiotic that may ensure the may preserve probiotic vitality and prolong their shelf life until use. So aim of our work was to develop a sort of bandage or patch as delivery system of live probiotics and to test their antibacterial activity against bacterial strains isolated from infected wound. So briefly, the method developed in collaboration with Prof. Serena Danti from the University of Pisa. We optimized a custom-made bio ink, and we mixed the bio-ink with the commercial formulation of probiotics containing several probiotic strains, and the mixture was then printed with next generation bioprinting to obtain the probiotic-laden patches. As you can see here, the bio-ink demonstrated a good printability, and both hollow and solid geometries were printed and easily ended with the potential to adapt to different wound shape. Then we also demonstrated that the printing procedure had no major effect on probiotic vitality, and also the developed patches showed an evident antimicrobial effect in standard laboratory conditions. As you can see here, this clear zone all around the patches is indicative of bacterial inhibition that was demonstrated against several bacterial strains, E. coli, Staphylococcus aureus, and Pseudomonas aeruginosa, differently to what observed with the control patch, which were empty, without probiotics. But microbiology laboratory is quite different from the in vivo situation, where there are several factors that may hamper the activity of new antimicrobial strategies. So, for example, in vivo, bacteria tend to grow as aggregate called biofilms. There are also proteins such as fibronectin, collagen, which facilitate bacterial attachment, and there are host components that may hamper the activity of antimicrobials. So the next step was to test the probiotic-laden patches in an in vivo-like wound infection model. And briefly, the model consists in a matrix of collagen type 1, which is the main component of the dermal layer in the skin, which was used as substrate for the biofilm formation. And then everything is included in an artificial liquid, which is a simulant wound fluid, which mimics the composition of the wound inflammatory exudate. So we developed the model, and then we infected with bacteria. We used Pseudomonas aeruginosa, which is one of the main wound pathogens. And once the biofilm was formed, we treated with the probiotic-laden patches. And after 24 hours of incubation in vitro, we quantified the number of survivor Pseudomonas as compared to the untreated samples. And as you can see here in this part of the graphic, the patch-treated samples showed a marked decrease in the biofilm-associated bacteria, around 2.5 log, which correspond to more than 99% in the number of viable bacteria as compared to the untreated samples. But this was obtained only when we supplemented the medium with glucose as supplementary nutritional component. So the systems look promising, but of course it needs further optimization. The second example is the use of postbiotic for the treatment of respiratory infections. In this case, we obtain postbiotic from a common probiotic, Lactobacillus rhamnosus, with a simple procedure. I will not enter into detail. And we use this postbiotic in an air-liquid interface lung infection model to mimic the condition found in the lung epithelium. In this case, the model briefly consists in a layer of epithelial cells directly in contact with air. Scusate un attimino. Okay. Directly in contact with air, and 8-10 days of culture in the presence of a differentiation medium, the monolayer differentiate in a polarized monolayer with morphological and functional feature of the in vivo airways, as you can see in this scanning electron microscopy image, where, for example, is clearly visible the ciliation of the epithelium. Then we infected the model with the bacteria. Again, we use Pseudomonas aeruginosa, as it is also one of the major respiratory pathogens, and the bacterium showed the ability to grow well in the model, and so we treated the infected monolayer with the postbiotics. So here are the main results obtained. We observed a strong anti-pseudomonal activity of the postbiotic with a reduction of 3 logs, which means more than 99% of the live P. aeruginosa in the model. This result was also confirmed by scanning electron microscopy. As you can see here, in the untreated monolayer infected with Pseudomonas, these bacterial aggregates were visible, resembling the early stage of biofilm formation, while these aggregates were virtually not visible in the infected and treated samples with postbiotic. The active concentration were not toxic for the host cells, and importantly, the postbiotic did not induce resistance following multiple passages at subinhibitory concentration, as unlikely, for example, a commercial antibiotics, ciprofloxacin, which rapidly induce resistance against both P. aeruginosa and Staphylococcus aureus. So we were encouraged by these promising results, and we further evaluated the translational potential of our postbiotic, and in collaboration with Prof. Anna Maria Piras from the Department of Pharmacy of the University of Pisa, we developed a liquid formulation of L. rhamnosus postbiotic for aerosol administration, and we studied the aerodynamic distribution to the lung in an air in vitro model of the pulmonary system. To do this, we use a next generation impactor, which is a device acquired within the THE project. Professor Piras yesterday described the instrument within the Spoke 4. And briefly, it consists in a device connected with a breathing simulator, and it consists in several cuffs, which correspond to the several stages from 1 to 7, which in turn correspond to the different zone of the airways, the upper, the central, and the lower airways. So the results obtained demonstrated that more than 60% of the aerolized postbiotic were able to reach the stage 4, 5, and 6 of the device, and they were made of fine particles with a mass median aerodynamic diameter less than 5 micron, which is ideal for the for reaching the deep airway. So, in conclusion, we demonstrated the promising antibacterial activity of live probiotic embedded in a 3D printed patches, and in condition closely resembling the wound bed. And also we demonstrate in an early lung infection model that L. rhamnosus postbiotics were effective in their antibacterial activity at non-toxic concentration and their potential to reach the deeper regions of the lung. So ongoing studies are addressed as stability studies, fractionation of postbiotics to identify the active components, optimization of the delivery system, and also the study of the anti-inflammatory potential, and hopefully quite soon in vivo testing in the mouse model. I would like to thank all the people in mine and also in other groups involved in the UNIP Spoke 7 sub-project 5. And also I would like Luisa Bracci, the coordinator of the Spoke 7, and all of you for your kind attention.
Thank you very much. Any questions? No, so thank you. Thank you very much. Now this session is over. Actually, we will start right away the next one. So thank you very much. And thank you, Luisa, for co-chairing with me.